In this video, we want to look at one more new topic, RNA, ribonucleic acid. RNA is a polymer of ribonucleotide. That means one pentose sugar along with nitrogen base and phosphoric acid. Here pentose sugar is ribose sugar. Two functional group it have. Major role of RNA is to participate in protein synthesis. No RNA means no protein synthesis. Three classes of RNA molecule exist. What are the molecules? mRNA, tRNA or RNA, messenger RNA, transfer RNA, ribosomal RNA. It is a single standard molecules. RNA are single standard molecules. First one is mRNA. It record the information for any protein would be synthesis is an DNA. DNA in the nucleus. Where it is present? It is presented in nucleus. The protein synthesis structure ribosome in cytoplasm. Where protein synthesis will occur? Ribosome here in cytoplasm. So either DNA has to come out of the nucleus or ribosome should go inside. Both of these are not possible. So both of these molecules which can bring the code for DNA to ribosome. Where ribosome present in the cytoplasm, that job is done by mRNA. DNA is presented in nucleus, it have protein molecules, but protein synthesis will occur in ribosome, it is presented in cytoplasm. So here mRNA acts as a messenger. Code mRNA is synthesis on DNA. From DNA to mRNA, the process is known as transcription. From DNA transcription, we get mRNA. This is not one-step reaction. How the process takes place? This one is the DNA. DNA is the double stranded one. Double stranded DNA molecules. One strand has code for protein synthesis, which we need. Another strand, this one is the coding strand. Another strand is called as template strand or complementary strand. This template strand copy all the information, that is coding information, because it involved in mRNA synthesis. This one is a DNA molecules first synthesis. First synthesis it will form synthesis hnRNA. hnRNA. It have two part, coding part and a non-coding part. Dark one is coding part, light one is the non-coding part. Why should have non-coding part? This should be removed. Splicing, basically cutting with the help of endonuclease enzyme. After cutting, what will get? Coding part and non-coding part. Coding part is called as exon, non-coding part is called as intron. Now using another enzyme lipase, only join that coding part, exon and exon has joined, only coding part. This coding part, can we call mRNA? We cannot, because highly unstable molecule. It should be stabilization which takes place. In stabilization, head and tail should be added. Cap is made up of methylated guanine. Tail is made up of polyadenine. This complete exon part, this one is the complete, that means coding part, joined by using cap and tail added. After capping and detailing. Now this structure becomes stable. So this one is called mRNA. Now we will study the detailed structure of functional mRNA. This one is the starting codon, this one is the stopping codon. Here functional group, this one is capping methylated guanine, this one is the tail, polyadenine. So starting codon, stop codon, middle part is the functional part. This one is non-coding part. Here this one is the non-coding part. This cap is methylated guanine, tail is polyadenine. This one is the functional mRNA. This is detailed structure of functional mRNA. Notes format of RNA molecules. So DNA to hnRNA by the process of transcription, then capping and detailing, we get mRNA. It involved in protein synthesis. Tail is made up of polyadenine, cap is made up of methylated guanine. This one is the detailed structure of functional mRNA.